So we are investigating a train crash. We have a patient that has been around a train crash where chlorine gas has come out of a train car, and she's a security guard and she is having some trouble breathing because the chlorine has gotten into her lungs. You can watch the whole episode or the whole scenario on Gizmos. Hey, biology students, this is Dr. B, and we are doing the lab on diffusion, the STEM case in Gizmos. If you want to log into Gizmos, you need to log into Clever, and then you need to get into Gizmos from Clever, and then hit diffusion, hit launch on diffusion. So patients are on the way to the hospital. We are going to use the progress bar to move forward and backward in this case. Let's click the next page here. We're expecting patients from the train wreck. One of the train cars was carrying chlorine gas, which is spread across the town. We can click on chlorine gas here, and because it's a vocabulary word, it will tell us what chlorine gas is. It's chlorine. It's often used for chlorination in pools. It's that smell you detect when you walk into a pool. It has been used as a chemical weapon because it's hard on lungs, and it can combine with other ions such as sodium to form salts such as sodium chloride. We'll find out what happens when it interacts with the water in your lungs. So chlorine damages people's lungs if they breathe it in. It causes swelling and liquid to build up in the lungs. Let's review the Diffusion Handbook before the patients arrive. So before we go to the Diffusion Handbook, let's look at the lab sheet here. We are looking for information in the handbook about what happens when you breathe in, what enters the body through the lungs, or when you breathe out, what leaves the body, and what are we going to attach to the hemoglobin protein. So be on the lookout for those first three questions as we go into the handbook. So here's our Diffusion Handbook. This is an alveoli. It's one of the tiny sacs in your lungs, and this is the capillary around that tiny air sac in your lungs. So when we breathe in, we get oxygen in, and when we breathe out, we get carbon dioxide out. I'll play that again. When we breathe in, we get oxygen. When we breathe out, we're getting rid of carbon dioxide. And so those are the answers to the first two questions on the lab sheet. Oxygen in, carbon dioxide out. Air moves in and out of the lungs through a set of tubes that make up the airways. We're going to travel to different parts of the airways here. The trachea is the tube that you can feel in the front of your neck here. It's kind of ridged with cartilage, so it doesn't collapse when you're trying to breathe. The bronchi are down in your lungs. The trachea branches into two directions, one for each lung. The alveoli, the bronchi divide into tiny, tiny, tiny tubes, and then those each go into a tiny air sac, and the alveoli are those tiny air sacs. They kind of look like onion bags, and the onion bag around it is capillaries, and capillaries are carrying the blood. So now we kind of know the structure of the lungs. Let's figure out a little bit more the function of the lungs. Remember, structure in biology always relates to the function. So a single alveoli is an alveolus. Here's a capillary, and there are red blood cells whose job is to carry oxygen in the human body, and they're zooming by the lungs. And that makes sense. We want all of our red blood cells to go into our lungs and fill up with oxygen and then travel around our body and drop it off like a UPS truck at all of our cells. So the red blood cells are sort of like the UPS, FedEx, Postal Service for oxygen in your body. Capillaries look red when they're viewed from a distance. Capillaries actually have no color. They look red because of the red blood cells moving inside the capillaries. Let's zoom in and check out a red blood cell here. They look like little kind of smushed donuts. You can zoom out here. You can notice there's two different colors here. There's bright red and there's kind of dark red. The dark red ones haven't picked up oxygen yet, and the bright red ones have. You can see that color difference again. The walls of the alveoli and capillaries are very thin. So you want the cell membrane on the alveolus to be very thin, and because it bumps up against the cell wall of capillaries, and you've got those two cell membranes, cell membrane of the capillary. You've got those two cell membranes to cross if you're oxygen. So here's the red blood cell. And we're going to put some oxygen on our hemoglobin. There we go. There's the answer to question 3. We just put oxygen on hemoglobin. So in the lung, gases diffuse across the respiratory membrane, and there are 3 things that make a big difference in how well oxygen gets into your blood. The diffusion distance, so you know that it takes longer to drive a further distance than a smaller distance. If you have to drive several cities away, it's going to take longer than if you just, go down the block to the grocery store. There's also concentration difference, how different concentrations in different parts of the lungs versus the blood, that can affect the rate of diffusion, and the surface area. If there's water in your alveoli or any kind of fluid, it covers up some of the surface area that is needed for oxygen to enter your blood. So let's talk about diffusion distance. The rate of diffusion depends on the distance molecules have to travel. This is called diffusion distance. So there's 2 membranes here. There's the membrane for the alveolus, that's your lungs, and the membrane for the capillary. Let's look, before we go too much further, let's look at our lab sheet. Number 4 says the respiratory membrane is made of the walls of the alveolus and the capillary. How does the structure of the membrane reflect its function? Each wall is made of single cells. These cells are very thin, which means the diffusion distance is very small. So the structure here, those very, very thin cell membranes and tiny cells, make it easy for oxygen to cross those thin cell membranes. So there's the cell membrane of the alveolus, and here's the cell membrane of the capillary. They're right next to each other, so oxygen doesn't have to go very far. So the respiratory membrane, the size of that membrane is equal to the diffusion distance. The smaller the diffusion distance, the faster oxygen is going to enter your blood. If those cell membranes, for some reason, puff up or swell up, it's going to be a further distance for oxygen to cross, and it's going to be very difficult for our patient to breathe. So let's look at, let's do a little lab here. On the differences, how the diffusion distance affects the time for oxygen to cross. Oops. So we'll measure how long it takes for the concentration of oxygen in the capillary to double. So here's the capillary. Here's where oxygen is coming in from the air, where you're breathing in air. And here's your blood. And we're looking for this oxygen concentration here to double. And we're going to time that. The distance here is 10 microns. And we're going to just collect this data in a chart. So oxygen concentration doubled. The first time it took 5 minutes and 19 seconds for the oxidation, for the concentration to double. Let's keep doing a couple more trials and see if that same thing happens. When we have a relatively small diffusion distance, we get a relatively quick time. So now we're going to increase the distance to 20 microns. We're going to swell up these membranes here. We're going to make them further apart. And it gives the oxygen, the distance gets longer for the oxygen to travel. So when we have this at 20 microns instead of 10, you can see that the membranes are larger here, and it takes the oxygen longer. It's taking 996 instead of 514. So again, a bigger difference is going to mean it takes longer for the oxygen to get over here. Let's go look at our results. So at 10 microns, our average was like 5 minutes and 32 seconds. The membrane, when we doubled the membrane thickness, we didn't quite double the time, but it took a lot longer. What effect does the diffusion distance have on the rate of diffusion of oxygen? Does it increase the diffusion distance? Does increasing the diffusion distance decrease the rate of diffusion? Does changing the diffusion distance have little effect? Or does increasing the diffusion distance increase the rate of diffusion? Well, let's look here. We have one diffusion distance that's small and one that's large. We have a small time here and we have a large time here. So when you have a large time, that means there's slower diffusion. So on the lab sheet, it says, what effect does the diffusion distance have on the rate of diffusion of oxygen? Make sure you mark this on your lab sheet as well as on the lab. And 6, we want to use this table here to explain our results. So not just do you have the correct result, but can you explain your results using this data? Look at the times here and look at the membrane thickness. This is 10, this is twice as thick at 20, and the times are different. Now let's look at concentration difference. So the difference in oxygen concentration of the alveolus is bigger than the capillary. This is an example of concentration difference. So this is 75. I don't know if that's parts per million or parts per thousand. And this is the measurement for blood entering the lungs. The difference here between these two numbers is a good thing because it helps diffusion happen. The concentration of blood entering the lungs does not change very much from its normal value of 45 millimeters of mercury. So let's see how the concentration difference if we have alveolar air, which has some oxygen in it, where we're breathing in oxygen. That's when a patient might be breathing 100% oxygen in the hospital. And down here, you might be at altitude where there's not as much oxygen in the air and there's less concentration difference. If you've ever gone up a mountain and kind of struggled to breathe a bit, you're having a concentration difference issue. So let's start with a concentration difference of 60 and 10, and let's see how that works. We're waiting until the oxygen concentration of the blood doubles. It took 7 minutes and 72 seconds here. Diffusion happens a lot more quickly in your real lungs. It'd probably be more like 7 seconds instead of... So this concentration difference, 60 versus 10 to start out with, is the lower concentration difference. We're going to go here and we're going to do 120 versus 10. Last time it was between like 7 and 9. This time, I'm guessing it might be a little bit more quick. Let's see when we do our trials here. So it's down to 5. 4 or something. Let's see those results. So when the concentration of difference is low, the average is 823. When the concentration difference is high, the number is smaller. It's 527. So what effect does the concentration difference have on the rate of diffusion of oxygen? Does decreasing the concentration difference increase the rate of diffusion? Does changing the concentration difference have little effect? Or does increasing the concentration difference increase the rate of diffusion? This is a small difference with a big time, so there's slow diffusion here. And this is a big difference with a smaller time. So you make your decision here. Use the data in the table to help support your answer. So there's a key concept here. In many textbooks, concentration differences are called concentration gradients, but that's incorrect. Concentration gradients depend both on the concentration difference and the diffusion distance. So there's difference and there's distance. The difference means there's different concentrations on both sides, and the distance means how far that oxygen has to go to cross both of those membranes, the alveolar membrane and the capillary membrane. So we can look at the concentration gradients using a graph. The concentration difference goes on the y-axis. So here's an example of a concentration difference. And the diffusion distance, how far that oxygen has to go, goes on the x-axis. We can add the data for the alveolus, that is the concentration in the alveolus, and the starting point for diffusion. We can now add the concentration in the capillary and the diffusion distance here. The line connecting these 2 points is the concentration gradient. Notice this line is nice and steep. It has a good steep slope. That leads to good breathing. In a healthy person, the concentration gradient is very steep. So you and I probably have a very big concentration gradient. The concentration gradient becomes shallow when the concentration difference is decreased or the diffusion distance is increased. Let's change the values for the concentration difference and diffusion distance to see if the effects of the concentration gradient. So let's change the diffusion distance. We'll swell your lungs up with you've either breathed some chlorine or you've got some kind of lung infection, maybe you have COVID. That really swells your lungs up and that makes it difficult to breathe. So the diffusion distance really increases when your lungs are swollen. And if your blood doesn't have much oxygen in it, compared to the air, you're not oxygenating yourself really well. The concentration difference goes down. So this is kind of a slope of someone who might be struggling to breathe with COVID, unfortunately. So this is healthy. This kind of thing, a shallow gradient, is unhealthy. There's a low concentration difference because if you're having a hard time getting oxygen across into your red blood cells, the concentration difference is going to go down. And if your lungs are swollen up, which is causing this problem, the diffusion distance will have gone up and you have this shallow gradient and it's very difficult to breathe. When compared to normal, what effect would a shallow concentration gradient have on the rate of diffusion? Well, you should be able to answer this one yourself because we know that unhealthy people have this shallow gradient. All right, we skipped a few, but we're going on to surface area here. And we're going to see what surface area in the lungs, liquids can affect the surface area available for diffusion. We can move the slider up here and you can see if your lungs are filling up with fluid for various reasons. Your lungs can have some fluid in them. One of the most common is pneumonia. And we can see, here's a balloon that's going to represent my alveolus. And you can see this balloon, the light is going right through it. And it doesn't have, it has all the surface available for diffusion. If we have a little water, I have a little water in my water balloon here. You can just see it right here. And I pass some light through that. You can see that the light goes right through most of the balloon, but it's having a hard time going through the water part down here. And oxygen has the same difficulty. Oxygen can pass through the walls of the lungs, but it has a harder time going through if there's fluid. You can dissolve oxygen in water. That's how fish breathe. They breathe the dissolved oxygen in water, but we're not fish, and we have a harder time getting oxygen out of water. Our lungs work differently than fish. Fish have gills, especially adapted for getting the oxygen out of water. We can't do that. We're used to breathing air. So let's look at surface area here. Let's do a little data collection on surface area. So this is 100% surface area in your lungs. This is the balloon that doesn't have any water in it. And these times are pretty low. They're around 5-6 minutes, maybe less. Let's go to 50%. Imagine 50% of your lungs are full of water. So all about up to here would be full of fluid in your alveoli. So you don't have as much surface area. You don't have as much cell membrane to get oxygen through because some of that cell membrane is taken up by fluid, which is mostly water. It's fluid coming out of your blood. Your lungs are basically leaking, or your capillaries are basically leaking some fluid into your lungs. It can happen at high altitudes. It can happen for a whole lot of reasons. If your lungs are somehow damaged, if you've got bacteria or viruses attacking your lung tissue. We can see with 100% surface area, the diffusion is faster. With less surface area, with more fluid in your lungs, the time is slower. So what effect does the surface area have on the rate of diffusion of oxygen? Decreasing the surface area increases the rate of diffusion. Changing the surface area has little effect on the rate of diffusion, or decreasing the surface area decreases the rate of diffusion? You can answer that one, and that is number 11 on your lab sheet. You can look at the data table here, and you can use this information to support your answer here in number 12. This is part 1 of the diffusion lab. You can look on YouTube for part 2 to finish up this lab.